Hello, everyone. Thanks for attending and thanks to the organizers. My name is Nathan Maines, and today I will describe how we've been using targeted proteomics to support the development of a model of the mouse macrophage TLR pathway. Laser pointer. Great. Okay, so in 2015, we published an article describing a model of the mouse macrophage chemotaxis signaling pathway. These are the ligands, the receptors, the heterotrimeric G proteins, PIP2 kinases, PIP3, the GEFs, and the small GTPases, activators, and some inhibitors. We targeted this pathway for quantification by mass spec. The colors indicate the copies per cell. And that's not the only information we need to do the simulations. To get the other information that we needed, we used two sets of microscopy data. A little bit of that data is illustrated up here. And then what we did was we manually trained a model of this pathway to reproduce in silico what we saw by microscopy. And that's depicted here. So each of these is a cross-section through the center of the macrophage in silico. And the color indicates activated RAC, which was used as a measure of chemotactic direction. So in this project, the goal is to develop a model of the TLR pathway to enable simulation at the molecular interaction level. So shown here is LPS from bacteria. It makes its way over to TLR4, which dimerizes at the cell surface. It will form a mitosome. At the endosome, it will form a tryphosome. And ultimately, this causes activation of the MAP kinase cascade, NF-kappa B, and IRFs. Now, to model this at the level of molecular interactions, we need two data sets. We need to know the abundance of the proteins, of each protein. So for doing that, we're doing targeted mass spec. We need to know the reaction rates, the rates of each interaction, the Caons, Chaos, and Kcats. And we're using molecular simulation for that. And to do the actual biological pathway modeling, we're using Simune. The mass spec is fairly straightforward. We prepared bone marrow-derived macrophages. We lysed and homogenized the sample. We spiked in heavy peptides, digested with trypsin, and performed LCMS on an Orbitrap system. The heavy peptides are quantified in femtomoles, nanomoles, and we spiked in a known amount. We measured the relative abundance of the heavy peptide to the light peptide that was originated from the BMDM protein. And so since we know the relative abundance and we know the abundance of the heavy peptide, we can calculate the abundance of the BMDM protein. We developed two peptides per protein, so we get two independent measurements. If they disagree, that's because we probably were unaware of some splicing or a PTM or a chemical artifact. And what we do is we develop a third or fourth or so on peptide assay to figure out what's really going on. So we've done 2 rounds of peptide assay development. We're in the middle of a 3rd round. Here's what we have so far. This is the core TLR4 pathway. Here's TLR4. This side is basically the NF-kappa B side. This side is the MAP kinase side. The nodes in color, this indicates the molecule abundance in copies per BMDM. The nodes that are white are nodes that we targeted and attempted to quantify, but so far we've failed. The most likely reason is because these proteins are below the detection sensitivity threshold of our LCMS mass spec. And the nodes in gray are ones that we want to target, but we haven't done so yet, but we will. A former postdoc in the lab did RNASeq of BMDMs grown up the same way we did. And so I took her data set and looked for a correlation by comparing it to our data set. And there was a correlation. It's R squared of 0.489, moderate correlation. And so in the presence of a successful protein abundance measurement by mass spec, we used that value for our model. But if it was a missing value, if we were unable to quantify it, what we did was we took the transcript abundance value and used this equation to predict the protein abundance value. And this should be accurate based on these values to roughly give or take a factor of 5. And so in this way, we were able to populate the entire core TLR4 pathway. The dash borders indicate that this is a rough estimate using the RNA-seq data. I mentioned at the beginning that we need 2 types of data. In addition to the protein abundance values, we need the Caons and Chaos and K-cats. The Caon range is depicted here. It's roughly a range over 1,000,000-fold. Depicted right here is the K-off range for signaling pathways. It's roughly a range of 10,000-fold. And so what we've done is we've used AlphaFold to create predicted structures of each of the TLR4 PPIs. And then what we did was we took those structures and inputted them to predict Caon rates using 2 programs, Simulation of Diffusional Association, or SDA, and Transcomp. SDA does have a feature where it can predict K-off rates, but they warn in the instructions, they're very clear that this feature is experimental and not at all validated. And we tested it using, from the literature, 20 PPIs unrelated to TLR4, 20 PPIs where we have published Caon and K-off from experiment and structure of the protein complex. And the K-off values, unfortunately, were not of any value, the predicted K-off values. They were off by many orders of magnitude. So we've only been predicting Caon values so far. AlphaFold has had a huge impact, and we have also seen really impressive results. We used AlphaFold 2 to predict the structure of the PPI complex. These AlphaFold predictions, you can see from the color, they overlap extremely well, and the AlphaFold scores are extremely high. These are above average. These are not every time is it this good, but oftentimes the structures are very good and can be used for the simulations. So this figure here is from Transcomp. It's from their publication. And what they did was, from the literature, they pulled out experimental Caons, and they downloaded the structures of the protein complexes, and they used their software to predict the Caon. And you can see that's accurate to, roughly speaking, give or take a factor of 5. So we took the TLR pathway PPIs, we ran them through AlphaFold, pulled out the structures, and then we did both SDA and Transcomp, and they agree somewhat, an R squared of 0.42. And so this is SDA simulating one of the standards. This isn't from the TLR4 pathway. This is one of the known structures. And you can see how it simulates. Both proteins are rigid. And this takes place over a period of roughly 1 microsecond in silico time. Okay, so now we have our set of measured protein abundance values and some predicted values based on RNA-seq, and we have some predicted Caon values. We don't have any predicted K-off values. And so now we're using Simune, and just for a little experiment, a little test, we modeled this tiny subpathway. So here's lipopolysaccharide from the bacterial cell wall. It binds to LBP and CD14, then MD2. Then MD2 bound to LPS will bind to monomeric TLR4, which will then dimerize. And so that's the end of this small pathway. They're not depicted, but there are 7 K-off rates associated with this tiny pathway. One of them is illustrated right here. In Simune, you don't need a background in bioinformatics or to write any differential equations. You just basically draw out each reaction, in this case the K-off reaction of dissociation. And I don't have any data for this, unfortunately, so I just put in 10 per second as just a very rough guess. I can just do that simulation, or I can tell Simune to sample K-off values from a distribution exploring the space around this value. And we can use a number of different distributions like a log normal or uniform or Gaussian. And I just, for what I'm going to show you, I just used uniform distributions. Okay, so I input into SDA that the LPS was 10 nanomolar, and that started the reaction. So this is the in silico time during each simulation. This is the concentration of dimeric TLR4. That's the end structure of this little pathway. Each blue curve is a simulation. I did 10,000 of them, so it filled this all in with blue. And then for each of these 7 graphs, these are the exploration of the K-off space. The y-axis is the cumulative distribution. The x-axis is the value of each parameter, each K-off. And so in blue here, these are all 10,000 simulations. And all of these 7 that I explored in parameter space, these were uniform distributions. So the CDF of a uniform distribution is just a diagonal line. And then what I did was I drew a little rectangle here. And so let's pretend that we know that in BMDMs, this is the amount of dimeric TLR4 at this time point, which happens to be about 5 minutes. And so that selected these red curves. And so for this parameter, the red and blue curve are basically on top. So this constraint in the experimental data did not constrain this K-off. But on the other hand, this red curve is constrained to the first 10% of the range that I explored. So in this way, we can use Simune to manually constrain the experimental outcomes, and then that will give us potentially constraints on the parameters, on the interaction rates, K on, K off, K cat, and also you can do the same thing for the abundance values, excuse me. This can be done manually, as I've shown here, and Simune is also capable of doing this in a highly automated fashion. So where we are at is we have a set of parameters. Some of them are fairly accurate measurements, and a lot of them are rough estimates. We can take this set of parameters, we can explore the parameter space, and we can send in thousands or millions of parameter sets into Simune. And then what we can do is we can plug in experimental data into Simune. So I mean by that, for example, this flow data. These are 2 proteins measured over a time course and over a dose response curve of the LPS TLR4 signaling pathway. So we can take this data, plug it into Simune. Simune can accept relative abundance values, and it can accept absolute abundance values. And then we can do thousands or millions of simulations, and we haven't done this yet, but we're hoping to successfully train, test, and validate a model of the TLR4 pathway. So in conclusion, we developed a mechanistic network of the TLR pathway and also of other cytosolic PRRs. We published this last year. Most of the TLR4 pathway was quantified by mass spec, but there were missing values, and so we used RNASeq to fill in those missing values with rough estimates. I did not show this, but in parallel to doing targeted proteomics, we're doing targeted phosphoproteomics. We're measuring the absolute abundance of copies per cell of different phosphorylated forms of the TLR4 proteins. These from basal cells, these can be used as input parameters in cells over a time course or dose response curve. They can be used to constrain the parameters. We used AlphaFold SDN transcomp to predict K-on values, and we are now trying to develop a pathway model. I just want to acknowledge the many, many people who contributed to this work, especially Anthony Armstrong, who did a ton of the SDA transcomp and AlphaFold, Pauline and Jessica, who did a ton of the bench work and mass spec, and Alexandra, who's PI, who ran the whole show. Thank you.
Any questions? Questions? Okay.
Could you elaborate on how you used AlphaFold 2 and transcompute to determine the K-on values?
Sure. So transcomp and SDA, both as input, only require the structure of the PPI protein complex. And it doesn't do docking or anything like that. It requires input of the full complex. So it needs you to tell it what the binding site is. It will do simulations basically where the structures are rigid. There's a little bit of ways of exploring it a little bit further than that, but basically you're constrained to a rigid. So it's not like molecular dynamics. It's not very flexible. And then the simulations in general are what would be in vitro simulations. It has limited ability to simulate what would really happen in a cell. So it's a good to get a starting point for parameter exploration.
Any other questions? Okay, I have one question. So you use Simune. So is it accepting structural data or?
Simune basically only requires all of the interactions to be drawn out as a pathway diagram, basically, and then all it requires really is the abundance values of each reactant and the Caons, Chaos, and K-cats for any reaction. And then it doesn't require any differential equation or anything like that. It doesn't require you to tell it what the time steps are. It will automatically do that. So you only need the biochemistry of the biological pathway.